Hello, good morning, and I hope everyone is doing very fine. I want to assume that you really had a very good week, and we are starting another week. We pray that we shall go through the week very well, just like we did for the previous one. Welcome to today's lesson, and once again, good morning. Isaac Baguma, as always, here to continue with you through this topic of tissue respiration. Last week, we looked at tissue respiration and we saw that it is that process of energy formation, process by which energy is formed within our bodies. Remember, we saw that energy in our bodies is formed in three main stages. We talked of glycolysis, we talked of the Krebs cycle, and the third stage is what we call the electron transport chain or the electron transport system. We noted that all these processes occur within the cell, but glycolysis occurs in the cytoplasm, Krebs cycle occurs within the matrix of the mitochondrion. Then the electron transport system occurs on stalked particles of the mitochondrion known as the oxysomes. So by the time we ended last time, we had seen that the process beginning with glycolysis has the end product there as pyruvate. We saw all those changes from the glucose molecule until we end up forming two molecules of pyruvate or pyruvic acid. In other words, from each molecule of glucose, we form two molecules of pyruvate. We further noted after glycolysis, if there is no oxygen, in absence of oxygen, anaerobic respiration proceeds, whereby fermentation now takes place. We saw that fermentation in plants, the pyruvate is converted into ethanol and carbon dioxide, while in animals it is converted into lactic acid. Therefore, we saw that we have fermentation which is of or giving lactic acid, or fermentation giving ethanol, or fermentation like in bacteria giving us the acetaldehyde. But in presence of oxygen, then the Krebs cycle proceeds, where we saw the changes before the Krebs cycle: pyruvate undergoing decarboxylation and dehydrogenation, forming acetyl coenzyme A that is fed into the Krebs cycle. Last time we ended when we had seen the Krebs cycle, and we saw that from the Krebs cycle there is formation of ATP directly. There is also formation of reduced nicotinamide adenine dinucleotide, NADH2. We also have reduction of FAD, so that we form reduced flavin adenine dinucleotide, that is FADH2. We saw that Krebs cycle also liberates carbon dioxide. Carbon dioxide is given off. So I think that is where we ended last time. When we had seen glycolysis, we had seen the Krebs cycle. But now, before we move to the last stage of energy formation, which is the electron transport system, let us look at the importance of the Krebs cycle. In other words, how important is the Krebs cycle to the body? We've noted that among the products of Krebs cycle, we have hydrogen atoms, which hydrogen atoms are taken up by NAD or FAD. In other words, they reduce NAD, they reduce FAD, so that the products are NADH2 and FADH2. We also have carbon dioxide formed as another product. Therefore, what is the importance of the Krebs cycle? When we are noting that it provides hydrogen atoms needed to yield energy from the oxidation of a glucose molecule. What do we mean? Remember we said that each NADH2 molecule later yields 3 ATP molecules, while each FADH2 later yields 2 ATP molecules. But the NADH2 and the FADH2 are formed as a result of hydrogen atoms getting added to them, whereby the hydrogen atoms reduce these compounds, forming the NADH2 and the FADH2. But later, the NADH2 and FADH2 liberate energy as a result of now losing the hydrogen atoms, like we shall see later. Meaning that these hydrogen atoms, many of them, are provided by the Krebs cycle. Then the NAD and FAD are now hydrogen carriers. But later, these hydrogen atoms from the Krebs cycle are the ones that bring about the formation of energy derived from the oxidation of a glucose molecule. Number 2, in the Krebs cycle, we saw a variety of intermediates within. You remember the alpha ketoglutarate, the citrate, the malate, the fumarate or the fumaric acid, just like we have seen in the Krebs cycle. When we look at those intermediates, we are talking of the citrate, alpha ketoglutarate, the succinate, the malate, the oxaloacetate. Some books talk about even fumaric acid as another intermediate. Now, those intermediates within the Krebs cycle are used to manufacture substances in the body like fatty acids, amino acids, and carotenoids. Those intermediates that we are seeing in the Krebs cycle, changing from one state to another, are important because they are used in the manufacture of fatty acids, amino acids, and the carotenoids. Fatty acids, these are the building blocks of lipids. Though we know lipids are made up of fatty acids and glycerol. Amino acids are important because they are building blocks for proteins, like we shall see. Then we also have the carotenoids, which are equally important, especially when we come to that process of photosynthesis. So that is the importance of the Krebs cycle. Now, if we have a comparison, if we make a comparison between the Krebs cycle and glycolysis, remember, glycolysis is the first stage, Krebs cycle is the second stage. Now, a comparison, in both there is formation of NADH2. I think we remember that in glycolysis, there was formation of ATP directly and formation of ATP indirectly as a result of formation of NADH2. The formation of ATP indirectly, we said NADH2 is generated and each liberates 3 ATP molecules. Meaning we get to see that in glycolysis, there is formation of NADH2. There is formation of NADH2. Even in Krebs cycle, there is formation of NADH2. In both, ATP is generated. Liberation of ATP directly in both. Then there is a reduction in the number of carbon atoms of organic compounds. There is a reduction in the number of carbon atoms of organic compounds. What do we mean by that? When we look at glycolysis, remember when we looked at glycolysis, we began with glucose, which is a 6-carbon compound. But we ended it with pyruvate, which is a 3-carbon compound. meaning from a 6-carbon compound to a 3-carbon compound, there is a reduction in the number of carbon atoms. Looking at the Krebs cycle, when we have citrate is a 6-carbon compound, but it is converted to alpha-ketoglutarate, which is a 5-carbon compound. Alpha-ketoglutarate to succinate, which is a 4-carbon compound. What does that mean? That in both glycolysis and the Krebs cycle, there is a reduction in the number of carbon atoms of organic compounds. Both glycolysis and Krebs cycle involve pyruvate, or pyruvate is involved. Remember, glycolysis, the product is pyruvate. For the Krebs cycle to proceed, there must be pyruvate. Though we've seen it must be decarboxylated, dehydrogenated to form acetyl that combines with coenzyme A to form acetyl-CoA, which gets into the Krebs cycle. Then both of them occur in living cells. Remember, respiration occurs in living cells. Then the differences between glycolysis and the Krebs cycle. In glycolysis, we saw that we basically have NADH2. We don't have FADH2. Therefore, FAD is only in Krebs cycle, but not in glycolysis. Only in Krebs, but not in glycolysis. In brief, in glycolysis, carbon dioxide is not formed, while in Krebs cycle, there is formation of carbon dioxide, liberation of carbon dioxide. In glycolysis, we didn't see any point where there is release of carbon dioxide. But in Krebs cycle, each point we had a change, like from a 6-carbon compound to 5-carbon compound, carbon dioxide liberated. From a 5-carbon compound to a 4-carbon compound, carbon dioxide was also liberated. Therefore, carbon dioxide is liberated in the Krebs cycle, unlike in glycolysis. Where does glycolysis occur? In the cytoplasm. Where does the Krebs cycle occur? In the mitochondrial matrix. Glycolysis does not necessarily depend on oxygen, while the Krebs cycle depends on oxygen availability. Remember, the Krebs cycle will occur whether aerobic or anaerobic respiration, whether oxygen is available or whether it is not available. But Krebs cycle will not proceed in absence of oxygen. It will only occur. Therefore, it is dependent on oxygen. So those are the similarities and differences between glycolysis and the Krebs cycle. But remember, like we said, that respiration occurs in 3 stages: glycolysis, Krebs cycle. We now move to the last stage, and that is the electron transport system, or the electron transfer system. Meaning at this point, the electron transfer